Today our lecture 15 is going to be on basics of heredity. Have you ever looked at the family albums and see resemblance between members of the same family? The members of some families have long, slender faces, while the members of other families have round faces. Other physical traits are easily identified too. The traits a family shows are passed from one generation to the next. For many years, people believed that traits were passed to the next generation through the blood. Now we know that the chromosomes carry traits from one generation to the next. Heredity is the passing of traits from one generation to the next generation. Inheritance is receiving these traits from parents. Genetic is the study of heredity. Gregor Mendel is the founder of genetics. Between 1856 and 1863, he cultivated and tested some 28,000 plants, the majority of which were pea plants. His experiments strongly indicated a physical factor that determines heredity. The abbot, Johann Gregor Mendel, a member of Augustinian order in Brann, Moravia, is often depicted as a kindly old man of the clothes who, while pottering around his monastery garden, somehow stumbled into important genetic laws. Again, our tendency to embellish the memory of already worthy individuals has led us astray. The real Mendel was remarkable enough in his own right. In many ways, he seems more like a 20th century biologist, somehow displaced into the wrong century. His first published paper is a landmark in its clarity and a model modern experimental report. Many scientists feel that it is perhaps the best scientific paper ever written. Early in his life, Mendel began training himself, and he became a rather competent naturalist. To support himself during those years, he worked as a substitute high school science teacher. The professors at the school, noting his unusual abilities, suggested that he take a rigorous qualifying examination and become a regular member of the high school faculty. Mendel took the test and did reasonably well, but failed to qualify. The standards for high school teachers were very rigorous. Mendel joined monastic order. His monkish superiors, confident in his abilities, sent him in 1851 to the University of Vienna for 2 years of concentrated studies in science and mathematics. Here he learned about the infant science of statistics, which was to serve him well. When Mendel returned to the monastery, he began his plant breeding studies in earnest. He impressed his fellow monks with his intelligence and vigor, and he applied these qualities to study of plants breeding. He developed new varieties of fruits and vegetables, kept abreast in latest developments of his field, joined the local science club, and became active in community affairs. Mendel began to experiment in the effects of crossing different strains of the common garden pea. To begin with, Mendel based his research on very carefully planned series of experiments and, more importantly, on a statistical analysis of the results. The use of mathematics to describe biological phenomena was a new concept. Clearly, Mendel's 2 years at the University of Vienna had not been wasted. The careful planning that went into Mendel's work is reflected in his selection of the common garden pea in his experimental subject. There were several advantages in this choice. Pea plants were readily available and fairly easy to grow, and Mendel was able to purchase 34 true breeding strains. These strains different from each other in very pronounced way, so that there could be no problem in identifying the results of given experiment. 8 years study of inheritance in pea plants allowed Mendel to collect data, analyze them, and eventually explain the basic principles of heredity. In his experiments, Mendel chose 7 different pairs of traits to work with. One of them is a seed form, could be round or wrinkled. Another is a color of seed content or cotyledons, could be green or yellow. Color of unripe seed pods, green or yellow. Shape of ripe seed pods, inflated or constricted. Length of stem, long or short. Position of flowers, either axial along the stem or terminal at the end of the stem. Color of flowers, either purple or white. All 7 pairs of contrasting traits you can see on this table, and that is form of the seed, color of the cotyledon, colors of flowers, pods, they are form of the pods and the color of the pods, position of the flowers, now and length of the stem. So Mendel studied these 7 pairs of contrasting traits that are easily observed in pea plants. Mendel predicted the possible outcome of these crosses. He used probability or mathematics of chance to make his predictions. In some of his experiments, he used pure lines of traits. This pure lines refers to a plant and its offspring that shows a particular trait generation after generation. Pure lines were achieved by allowing plants to self-fertilize for several generations. When 2 different pure lines were crossed, a hybrid plant was produced. This hybrid generation of plants is called first filial generation, or F1 generation. F stands for fertility, not the other word. In one of his experiments, Mendel crossed a pure tall plants with a pure short pea plants. Mendel suspected that medium-sized plants would be produced. However, Mendel found that only tall plants appeared in the first filial generation. When Mendel experimented with similar crosses for all the pure lines, he got the same results. One trait was outwardly shown and the other traits seemed to disappear. Mendel then allowed hybrid plants from the first filial generation to self-fertilize. In the next generation of plants, the F2 generation, Mendel was surprised to find that both pure line traits appeared. That is, some of the new plants were tall, other plants were short. This meant that the trait that appeared to be lost in the first filial generation had reappeared in the second filial generation. To explain this unexpected result, Mendel hypothesized that factors control heredity and that these factors occur in contrasting pairs. For example, one factor controls a trait for tall pea plants and opposite control for the short pea plants. In analyzing his result, Mendel concluded that some traits were stronger or more assertive than others. He called the strong traits dominant traits and the weaker traits recessive traits. Mendel's law of dominance states that when a pure dominant and pure recessive trait are present, the dominant traits will show up in the hybrid organisms. But remember, the recessive traits do not disappear. It shows up again in the second filial generation. To make such predictions, we're going to use a Punnett squares. Punnett squares are diagrams geneticists use to show the possible genotypes of a particular genetic cross. Punnett squares are allowed to visualize the possible outcomes of a cross. It is named after Reginald Crandall Punnett, who devised the approach in 1905. And here's how we're going to do it. On outside, we put factors that come from the parents. You see? Like here, one will be recessive, comes from one parent, and another would be dominant, comes from another parent. By matching one factor with another, we will know the offspring factors. So outcome of a cross will be inside factors of offsprings. Actually, in nowadays, we do not refer to these letters actually as factors, but refer to them as the genotype. So on outside, what you see of the Punnett square is a parent genotype, and inside of the square, of this square, Punnett square, we see offspring genotype. When we look at one trait, for example, as we just did at the length of the stem of the pea plant, we use 4 boxes. But sometimes we could do dihybrid cross when we looked at 2 traits, for example, color of the cotyledons, here yellow and green, and shape of the cotyledons, smooth or wrinkled. Mendel's factors are now called genes. A gene is a section of DNA on a chromosome that control the production of specific trait or protein. Since chromosomes occur in pairs, as you could see on this diagram, genes occur in pairs as well. The term gene was introduced by a Danish botanist, plant physiologist, and geneticist Wilhelm Johansen in 1909. Each individual carries 2 gene variant alleles for each trait. Each half of the gene pair is called allele. These pairs of alleles is inherited from the parents, with each parent contributing 1 allele. Geneticists use letters to represent alleles, as you could see on this diagram. For example, a capital T could be used to represent gene for tall, and a small t could be used to represent a gene for short. So what are we looking here at? 2 different alleles, correct. One allele comes from one parent and another allele comes from another parent. The full human sequence of the genes was published in 2003, with the exception of 1% due to limitation in current technology. It is now known that the human genome contains roughly 3 billion base pairs and 20,500 genes. The Human Genome Project has contributed to identification of more than 1,800 disease genes. But what kind of a gene is it? You see, we use a term heterozygous to describe gene that has 2 different alleles for the trait. In our example, that would be capital T and lowercase t. This is a heterozygous hybrid, which means one allele is dominant and another allele is recessive. And what gene or trait here we are looking at? We call this homozygous. Homozygous is the term used to describe an organism or gene that has inherited two identical alleles and is pure for that trait. In our example, it is two capital T's, pure, tall. So everything in repetition. Let's summarize just what we just discussed. Mendel hypothesized that factors, we call now genes, control heredity. Factors or genes occur in contrasting pairs. For example, one factor controls the trait for tall, another for short. Mendel concluded that some traits were stronger or more recessive than others. The strong traits he called dominant, and the weaker traits he called recessive. How did he know which trait is dominant and which is recessive? You see, when he crossed two pure lines, if he crosses two pure lines, one, for example, always tall and another is always short, if in first filial generation appears the tall trait, that means that tall is a dominant trait. Now let's cross pea plants with smooth seeds and with the wrinkled one, pea plants with wrinkled ones. So if in the first generation, as we could see, appears the smooth one, that indicates that the smooth shape of the seed is dominant trait. In this case, the color purple is dominant in the flower. In this case, the seed pod that is green is dominant. So color green is dominant because that's what appears in first filial generation after two pure lines being crossed. Here we have inflated pea pod and constricted pea pod. As a result, we ending up in first filial generation with the inflated pea pod, seed pod. Now, what does it mean? It means that inflated trait of the seed pod is dominant. Here is examples when we cross pea plants with green seeds and yellow seeds, and the result in the first filial generation is yellow. So that means the color yellow is dominant trait. Here we're looking at the position of the flower at the terminal. Well, this is terminal position on the end of the stem, and this is axial position on the side of the stem. So we ending up in the first filial generation with the flowers on the sides of the stem. That means this trait is dominant. Mendel's law of dominance states that when pure dominant and a pure recessive traits are present, the dominant trait will show in the hybrid organism. But remember, the recessive trait does not disappear. It shows up again in the second filial generation. But so far, we have given only observations and demonstrations, not explanations. What really happens when two different alleles occur together? How is one suppressed? And how does the organism choose between two sets of information? Actually, there are several different ways that dominance can happen. The most usual case is simply that the recessive allele isn't doing anything. For instance, many genes make enzymes. An allele that isn't making anything will result in phenotype based on the absence of an enzyme's function. This is clearest in rare medical disorders, when the absence of an enzyme can have a severe and even lethal effect. In a relatively benign example, albinos lack an enzyme that is necessary to make melanin pigments. They didn't get functioning gene from their father and they didn't get one from their mother. So their cells cannot make the pigment. Such individuals are homozygous for recessive alleles, which in this case are alleles that are not functioning. Heterozygous for albinism or other enzyme deficiencies, on the other hand, have one working allele and one doesn't work and produce only half the usual amount of enzyme. But in most cases, half the normal amount of enzyme is still enough to metabolize all the enzyme substrate, and the phenotype of the heterozygote will be perfectly normal. Also, we have been talking about recessive alleles that do not do anything. The same situation occurs when the recessive allele is simply doing less of something. Such alleles are still be masked by more active dominant partners. Sometimes dominance is in the eye of the beholder. For instance, many mutant genes have been found in creating bizarre changes in the eye color over fruit flies. Almost all of them are recessive to the normal alleles, but are involved in the synthesis of one or both of two eye color pigments. For some of the gene loci involved, dominance is once again just a matter of half enough enzymes being enough to complete some necessary step. For other genes, however, the situation is different. Even in this, the recessive mutant allele is not functioning, but here half as much gene actually results in exactly half as much eye pigment being produced. This can be determined easily enough through the pigments are extracted and quantified. But in the living fly, the observer cannot tell the difference between a fly with its full normal amount of eye pigment and the heterozygote with only half as much pigment. And so the normal allele is recorded as being dominant. It is as though three genotypes were represented by a jar full of India ink, a jar full of water, and jar with 50-50 mixed of ink and water. All one can see is that there are two jars with black fluid and one with clear fluid. There is an important lesson here. Dominance depends on how the genotype are classified. In some cases, the dominant and recessive interact in another way. The recessive allele is involved in some normal function, and the dominant allele makes some kind of gene product that prevents that function. Today we know that there are times when neither trait is dominant over others. This condition is called incomplete or partial dominance. When this situation occurs, two opposite traits are combined and a blending traits appears to happen. For example, if a flower is incompletely dominant for color and the red flower is crossed with white flower, a pink flower results. However, in second filial generation, the original traits reappear. When two pink hybrids for flowers are crossed, 1/4 of the second filial generation will be white, 1/4 of this generation will be red, and 1/2 will be pink. We can represent these crosses as follows. Here, red flowers will cross with white. We ending up in the first filial generation with pink flowers. In second filial generation, we have red, white, and pink. Half of them are pink, and quarter white, quarter red. Or it's even better, we can use Punnett squares that we learned in the beginning of this lecture how to use. So first we put the parent's genome on outside of the Punnett square, and then we matching up each of the alleles of the parents. One comes from one plant and another comes from another one. This is a filial generation cross. Now, second filial generation cross, we're using the same Punnett squares. right? But here we already have pink flowers. We cross them, and we ending up with the following ratios. Half pink, quarter red, and quarter white. More to make it more visual, we can use pictures. While Mendel's work laid the foundation, it was German botanist Karl Correns who is credited with the actual discovery of incomplete dominance. Correns conducted his plants research in early 1900s. That is when he observed a blend of colors in flower petals. He used 4 o'clock plants. In our examples, we use carnations. From his data gathered in the second filial generation, Mendel proposed the law of segregation. This principle said that each individual carries 2 factors for each trait. This pair of factors is inherited from the parent, with each parent contributing 1 factor. The individual's factors segregate during meiosis, so the 1 factor ends up in each sperm or egg cell. Modern interpretation of this law. Alleles of a gene separate into equal numbers of gametes, or sex cells we call it, right? Now, allele is an alternative form of a gene. Dominant is an allele that masks expression of another allele, and recessive is an allele whose expression is masked by another allele. Genotype describes the actual makeup of an individual organism. It describes the combination of alleles. For example, if tall is dominant over short, a tall pea plant could be represented as capital T, or capital T and lowercase t. Either combination of genes will produce a tall plant. A short plant would be 2 lowercase t's. Only this combination of 2 genes for shortness will produce a small plant. Genotype describes the actual makeup of an individual organism. It describes the combination of alleles. For example, if tall is dominant over short, a tall pea plant could be represented as capital T, or capital T and lowercase t. Either combination of genes will produce a tall plant. A short plant would be 2 lowercase t's. Only this combination of 2 genes for shortness will produce a small plant. Genotypes allows us to recognize and express traits that are inherited from the parents. When a couple undergoes genetic testings prior to having a baby, a scientist must determine the couple's genotype. This allows the parent to find out if they have genes for diseases such as sickle cell anemia or hemophilia or Tay-Sachs. Because height in humans is determined by multiple gene variants, an inheritance pattern called a polygenic inheritance, it is difficult to accurately predict how tall a child might be. So it is not like in plants. In plants, height is a simple trait. In humans and animals, height is a complex trait that is determined by not 1, but many genes. So the genotype will be a combination of letters which represent the genes. Now let's go back to the Mendel's law of segregation, especially if you didn't understand it at the first time. Now, the individual's factors segregate during meiosis so that 1 factor ends up in each sperm or egg. So here I'm showing it to you visually what happens. Segregation here happens. You see, we have 1 gene here, right? That is heterozygous, right? and the segregation occurs during meiosis, and we ending up with two sperms that each will carry the allele from that gene. Doesn't matter what kind of gene it is, what it codes for, right? Either heterozygous, homozygous dominant, or homozygous recessive. And exactly the same thing happens in females when the eggs are produced during oogenesis, the process called oogenesis, right, during meiosis. So why genes segregate? Because chromosomes segregate during meiosis. As you remember, XX is a genetic makeup of a female, right? On sex chromosomes, if there is XX, the female will be born. Now we have XY, the male is going to be born. Now, but then eventually they grow, right? They produce sex cells during meiosis, and look what happens. Then each of the chromosomes segregates and ends up in different eggs. Here we see happening, it's in females. We know this process as a formation of egg is oogenesis. And here spermatogenesis in males happens, and each of the chromosomes ends up in a different sex cell or sperm cell, we call it, right? Now, looking at these diagrams, please answer the question. Who determines the sex of the child? Mother or father? Of course, the father will determine the sex of the offspring from what you see on this diagram, right? Because females do not have Y chromosomes. And it depends what type of sperm is going to fertilize an egg. If it's going to be an X sperm, there will be a girl born, and if there will be a Y sperm fertilizing the egg, there will be a boy born. So you understand how ridiculous it is when some man says, what is wrong with my wife? She cannot give me a birth to a boy. I have all girls, right? So who determines the sex of the children? Father, of course, not mother. So from what you see, females produce one kind of eggs, and males produce two different types of sperm cells. Some of them carry X chromosomes, while others carry Y chromosome. Another important term in genetics is phenotype. Phenotype describes the outward expression of the genotypes. When you look at these boys, you see different genotypes, different phenotypes that determined by their genotype, and therefore you see different color of the eyes, different shape of the eyebrows, different colors of the hair, different shape of the lips, and so on. That is what we refer to phenotypes. Phenotype in some instances also can be affected by the environment. And on this slide, you see the genotype. That's how we write genotype, by the letters, right? Each gene represents two alleles, one that comes from mother and one that comes from father. What you see on this slide is the different phenotypes of the nose and ears. Noses and ears. Here you could see a Roman nose. This type of nose is characterized by a prominent bridge, which makes it appear bent, and in sloping curve protruding from the face. The next to it is a straight nose. Here, look at the ears. This ear has an attached earlobe, and this ear has a free earlobe. This is what we call phenotypes, outward expression of genotype. Knowing that Roman nose is the dominant trait, we can predict its genotype. It would be either a homozygous dominant or heterozygous? Straight nose is a recessive trait. So what would be its genotype? Think about it. You are correct if you say this is homozygous recessive. Same thing with a attached earlobe. So it is homozygous recessive as well. While free earlobe is a dominant trait, therefore the genotype is going to be either a homozygous dominant or heterozygous. So what we just looked at, different phenotypes and genotypes, and we saw how genotypes can determine the outward expression of phenotypes. Let's take a look at the different phenotypes and case scenario. You heard of Tay-Sachs disease when we discuss lysosomes. If you don't remember, I remind you, Tay-Sachs disease is a rare genetic disorder passed from parents to child. It's caused by absence of an enzyme that helps break down fatty acids. So there is no gene that actually responsible for production of this enzyme. These fatty substances, they build up to toxic levels in the brain and spinal cord and affect the function of the nerve cells. The babies are born and are like normal in the first several months of life, and then they start to lose motor skills and go downhill neurologically over a period of months and usually die within a few years of diagnosis. Now, this disease is determined by a recessive gene. Let's abbreviate it as lowercase r. Other examples of autosomal recessive disorders include cystic fibrosis that we also discussed on previous lectures and sickle cell anemia. So if we have two parents, father and mother, that are heterozygous on the gene of Tay-Sachs disease, what their chances of having offspring with the disease, Tay-Sachs disease? Let's draw a Punnett square. First we will put mother's genotype, then we put father's genotype, and then we're going to match alleles, which will represent genotype of the offspring. So here we put fathers, mothers we match, fathers, mothers, then we match again fathers, mothers. We always first dominant allele first we put. Now then we match fathers and mothers. So what did we end up with? What is genotype ratio? 1:1. That would be a genotype ratio. We're going to have two, children that will be carriers. One would be normal and one will be what? Carrying the Tay-Sachs disease, or afflicted, other way to say it. Now we're looking at the genotypes, percentages. So 25% will be normal, 50% will be carriers, and 25 are going to be afflicted. Phenotype ratio, we can put one normal, two carriers, and one afflicted. So phenotype percentage obviously are going to be 25 normal, 50 carriers, and 25 afflicted. Now we're going to look at the Mendel's law of independent assortment. Just read into the phrase independent assortment. What does it mean? An analysis of Mendel's results showed that factors for different traits were inherited independently. Mendel called this the law of independent assortment. Here's modern interpretation of this law. Traits are inherited independently because the alleles of one gene are distributed to gametes independently. which is correct, right? It's what it means actually, that plants with yellow flowers not necessarily are all tall ones, right? Two different traits do not inherit it together. They inherit independently. And doesn't mean that plants, short plants, all will say seeds, smooth seeds. Again, the traits inherited independently. They do not depend one on another. Therefore, he called it law of independent assortment. But in science, there is always an exception. So we're going to look at the work of the Thomas Hunt Morgan at Columbia University. In 1910, he began to apply Mendel's techniques, but instead of breeding pea plants, he began program of breeding experiments with the fruit flies, with Drosophila. To most people, fruit flies are just pests that hover over the overripe bananas, leftover apples, or other forgotten fruits. However, to scientists, fruit flies are an important organism, and understanding what goes on within these tiny bugs can reveal big things about basics of biology. But before going any further, let's answer the question, why had Drosophila turned out to be a perfect model in the study of genetics? And here's reason number one. They are easy to maintain in the laboratory. They mature in 10 days. The males could easily be told from the females, as you could see from this diagram. They mate readily. Each female lays hundreds of eggs. They can be anesthetized for inspection. I should say easily anesthetized for inspection. They have only 4 chromosomes, which makes it easy to analyze them. Their mutants are readily recognized. Wild type, brick red eyes, and mutants, white eyes. You can see it from this slide. Here you could see a wild type bricked eyes and mutants white eyes. Morgan's laboratory became known as the fly room and the scientific team as the fly squad. Morgan found that the eyes color of Drosophila is linked to its gender. The X chromosome. Morgan had proven that genes are on chromosomes, and that he showed the general concept of gene linkage. He discovered X-linked inheritance. So what is the X-linked inheritance? First, let me remind you that chromosomes come in pairs. Genes are located on the chromosomes. For each chromosome pair, one comes from each parent. Both have the same genes arranged in the same order, but with a slight variation in their DNA sequences. The chromosomes are very long strands of DNA coiled up like a ball of string. Genes located on sex chromosomes are called sex-linked genes. They tend to be inherited together because they are located on the same chromosome. Such genes are called linked genes. Since a male has only one copy of an X-linked gene, Y chromosome behaves as if it has a recessive allele for all X chromosome loci. Loci are particular position, point, or place. Some people say loci because in English, C is read as before I read as a C. But classical way to read it is loci. So we will keep calling it loci. Locus is singular, loci is plural. For X-linked genes in males, the terms hemizygous. Hemi means half, as used instead of homozygous or heterozygous. Knowing this, let's start thinking and answer a question: how many recessive alleles must a female have in order for a recessive trait to be expressed? The answer is 2. Now try to explain it. In order for a trait, a disorder or condition to appear, therefore, there has to be 2 of the alleles are present on both X chromosomes. In females, like for example, there is such conditions in humans as a color blindness. There is more colorblind males than females. Why? Because colorblind, for a colorblind to appear in a female, it is a condition that associated with sex chromosomes, right? X chromosomes. It has to be on both X chromosomes. The same thing with the baldness. There is more bald men than females. Why? Again, because in order for baldness to show up on the females, the trait has to be present on both X chromosomes. However, in males, if the trait is, if the allele is present for a certain condition, there is no next X chromosome to the X chromosome, so there is nothing to mask it. There is very few genes on Y chromosome. So therefore, the more men, there is more people that are bald, or people that are colorblind, or males that are colorblind, right? Because only one allele is necessary for condition to appear. Same thing with hemophilia. Hemophilia is known as the bleeder's disease, when the blood doesn't clot normally. Most hemophiliac males formally bleed to death in their youth, but modern medicine has allowed afflicted hemophiliacs to survive and reproduce through the transfusion of blood-derived substance known as anti-hemophiliac factor that supplies the critical substance missing in hemophiliacs. Let's summarize what are common X-linked conditions in humans. Hemophilia, red-green color blindness, between 7 and 10% of men and 0.49 to 1% of women are affected. Its commonness may be explained by its relatively benign nature. Another X-linked condition is androgenic alopecia. Androgenic alopecia is a male pattern of baldness that is passed from mother to child. A man can more accurately predict his chances of developing a male pattern of baldness by observing his mother's father than by looking at his own father. Mendel had begun the groundwork for the modern science of genetics. He correctly hypothesized and performed accurate experiments that supported his theories. Unfortunately, most of Mendel's work was not discovered until 1900s, several years after his death. He never knew the impact his work would have on society. We now use his theories to determine the probability of inherited disease and birth defects, and even to produce new and improved varieties of plants and animals. Mendel and other early geneticists provided the groundwork for our modern study of heredity. Today, we are even able to understand the chemical basis of heredity and use that information to study human inheritance. The application of genetics has become an important scientific tool in our constant search to improve our quality of life. Today in our brief lecture, we defined heredity and inheritance. We briefly reviewed the Mendel's experiments and took a quick peek into basics of inheritance by learning how to use Punnett squares. We learned about 3 Mendel's law, and that is law of dominance, law of segregation, and law of independent assortment. We picked into incomplete dominance. And we learn about sex-linked genes, how some condition can be passed from parents to their offspring, in particular such conditions as hemophilia, red-green color blindness, and androgenic alopecia.